Hey there, greetings to you. This is the part 2 of our series of classes under the topic cell. Part 1, we introduced the parts of the cell. We said the cell is made up of three major parts, that is the cell membrane, the cytoplasm, and then the nucleus. And we saw the different levels where we can undertake the study of cells. That is a term that we introduced as cytology. We said we can study cells at a molecular level where we can find molecules arranged in atomic structures. We can study cells at macromolecular level where we can find the macromolecules such as proteins, lipids, carbohydrates, vitamins, and we said that level is usually biochemistry, and then the other first level, the level of molecules, is usually the field of science known as molecular biology. And later on, we can study cells at the level of tissues, and that study we didn't hint about it, but it is called histology, studying cells at tissue level. That is histology. Seeing or identifying out the abnormalities in tissue arrangement of those cells in the different tissues, or the function of different tissues. So cytology can be uh, approached in those different sectors of science or fields of science. This part we are interested in understanding the cell membrane, which we defined as a physical barrier between the intracellular components and then the extracellular environment surrounding our cell. So the understanding about that physical barrier, which is the cell membrane. And to start off our discussion about the composition of the cell membrane. The cell membrane is mainly composed of two major macromolecules, that is the lipids and the proteins. So to start off with the lipids as our first, in the cell membrane, we find several classes of lipids as part of the components within the cell membrane. So we find phospholipids, and then we find the cholesterol molecules, phospholipids, cholesterol molecules, and then glycolipids. So the three major classes of lipids that we find as components of the cell membrane. And they are not there as for decoration, but they play major or key roles that we are going to know about in this discussion. Uh, you should note that phospholipids in the plasma membrane, they are arranged in forms of heads and tails in a complex structure known as lipid bilayer. So they are arranged as heads and tails, but it is a bilayer. So we have one layer of an arrangement of head and tails, and then the other layer of an arrangement of head and tails. One layer is facing the cytoplasm side, and then the other layer is facing the uh, external surrounding environment to this cell. So that is the bilayer we are talking about. So the other molecules, lipid molecules that we find, be cholesterol and then glycolipids. They are carbohydrates. Okay, these the glycolipids are lipid molecules linked up to carbohydrate. So they end up with that name, uh, a glycolipid. So they have a carbohydrate part, then a lipid part. They are part of the glycocalyx, and we're going to see what the glycocalyx is. So this is our uh, cell membrane. Cell membrane is a lipid bilayer, phospholipid bilayer. The phospholipids there are arranged in the bilayer, and this is the bilayer here. You can see these pale yellow thread-like structures. So these are the tails, and then these purple structures here, like beads, they are the heads of the phospholipid molecules. So these are the phospholipid heads, and then these are the phospholipid tails of this cell membrane. So this is one layer facing the external side, and then this intracellular side we have this layer, heads and tails facing the internal side. So in the end we have two layers, two phospholipid layers, and we're calling those the phospholipid altogether a phospholipid bilayer. So it is a bilayer of phospholipids, this cell membrane. In addition, we also find cholesterol molecules. So they are here, the cholesterol molecules trying to link up these phospholipid bilayers. Uh, so they aid in making this membrane, this cell membrane, semi-permeable in nature. So can allow in or refuse something to enter the cell or leave the cell at a particular moment. So those are the cholesterol molecules within, and they are embedded within this phospholipid bilayer molecules, lipid molecules. In addition, we also have the glycolipids, and we gave a site where we find the glycolipids, and we said they are found in the glycocalyx site. So we have a glycocalyx here. So it is a glycolipid molecule attached to protein molecule, but this protein molecule is embedded within this phospholipid bilayer. So these ones can function in communication between cells, between body cells. So there's an adjacent cell that can be somewhere here, so they can always communication between this cell and an adjacent cell somewhere here. Or if this was a layer of white, if this was the cell membrane of a white blood cell, it can always signal another white blood cell adjacent to it that, hey, I have something infecting my internal area. So it sends off signals. So these glycolipid molecules can help cells in communication. So we also have uh, glycolipids, other glycolipid forms somewhere here, another one here, and then another one here. So those are the molecules, the cholesterol molecules within the cell membrane, the phospholipid bilayer molecules, and then the glycolipids. You can also see some proteins. We are calling these integral proteins somewhere here. Then this one we're calling it the peripheral protein. Facing to the external side, and then another peripheral protein facing the internal side. There is reason for that. And then we are kind of seeing a channel somewhere within this protein. So some of the proteins are uh, channels, transportation channels, but these proteins are located or embedded within the plasma membrane of the cell. So the other category of molecules that we find within the cell membrane are the proteins, and we can find them as integral proteins, peripheral proteins, or enzymes, ion channels, receptors, or we can find them as glycoproteins. So still glycoproteins can also function in communication between cells. And then also the receptors are protein molecules that appear as receptors on the surface of the cell membrane also function in communication between cells, or as you hear the word receptor is a hidden word there, which is to receive. So they can, most of them function as receivers of some molecules from the surrounding environment. So when this receptor receives, binds a particular molecule, then that molecule can be allowed uh, inside the intracellular components of the cell. So in the plasma membrane, we additionally have can find their proteins in terms of enzymes. So those ones can be functioning in modifying certain molecules that these cells have to receive, and then modifying that molecule at the cell membrane, and then that cell membrane is made ready for reception of such a molecule. So those enzymes that we can find on the cell membrane or embedded within the cell membrane, they function in such ways. You first try to modify whichever molecule that is coming, and then the molecule that has been modified can be allowed in by this plasma membrane. Remember, I said plasma membrane is mostly composed of phospholipid bilayer. So the molecule cannot interact with the phospholipids, which are the major component of the plasma membrane. Then these enzymes can be functioning for such a purpose. First, modify this molecule that is requesting for reception within the cell, and then is modified, and the receptors bind it, and then is taken up into the cell. Other proteins function as ion channels. So we have a channel in the cell membrane, and then that channel is where a particular molecule will have to pass. So it is prevented from interacting directly with the phospholipid molecules that we saw in the phospholipid bilayer earlier on. So that prevention of interacting with the phospholipid bilayer molecules helps this particular molecule that is trying to request for reception in this cell be allowed to enter into the intracellular components by using this ion channel. So we are seeing the word ion. So those ion channels apply these, they function in allowing in or allowing out particular types of ions within or outside of this cell. So we also have peripheral membrane proteins, so those ones also function in receiving certain types of molecules still, but on the peripheral level. So this time the molecule will not utilize a channel to enter, but it will interact with this peripheral protein, make chemical bonding, a particular type of chemical bonding between the two parties, the molecule trying to receive reception, and then the peripheral protein. So they interact, and then this molecule is transported across the cell membrane to enter the intracellular compartment. Integral proteins also usually are found functioning in transportation, so it is there to receive particular molecules from inside the cell so that we transport it outside of the cell, or receive from outside of the cell and then transport inside of the cell. So these are the molecules that we are trying to discuss. We have glycoproteins here, so it is a protein linked to a carbohydrate molecule, so it has a carbohydrate attached to it. Then we have glycolipid that we saw earlier on. So the green ones here are the glycolipids, the blue ones are the glycoproteins. We have an integral membrane protein. So integral means kind of is kind of embedded across the whole membrane. So it peruses through both layers of the phospholipid, uh, both phospholipid bilayers. So it can receive something from the extracellular side, and then this something is allowed to pass through this membrane, through this integral protein to the inside. But we can have peripheral proteins only on one side, that is the internal or the external. Here I'm not seeing another peripheral protein in the external, but is you can see this one facing the internal side. So peripheral protein that can interact with something from the internal side for particular purposes. Maybe it is a peripheral protein but an enzyme in nature, so it interacts with particular molecules to be uh, modified for transportation, maybe by integral proteins or channel proteins. So we can have peripheral proteins on either sides, the external side and then the internal side. So those are the peripheral proteins, the integral proteins, and then which other types of proteins? The channel proteins here. So they are the channels where something can pass so that it is received inside or taken outside of the cell. So that is the cell membrane. So the functions of the cell membrane are it is selectively permeable in nature. So in its being a selective membrane, it allows in certain types of molecules to enter the cell or leave the cell. So if we are looking, uh, if we are trying to study the function of the cell membrane in terms of nutrients, particular nutrients that are supposed to enable the cell carry out its biochemical processes will be allowed in selectively. And then the molecules that are inside of the cell and we are considering them a waste material will be taken out selectively still by this cell in different transportation ways as we are going to see. So it is selective in its function as a barrier. So the other function of cell membrane is communication between cells, adjacent cells, communication with adjacent cells, or communication with certain extracellular molecules that are trying to be circulated. And that is done by the glycoproteins or the other glycolipids. So the cellular connections, yeah, certain tissues, because we know tissues are groups of cells that groups of similar cells performing a particular specific function. So those cells have to interact with one another, and that interaction we are calling it an intercellular connection. So between neighboring cells, there should be a connection. So this tissue will not be made up of cells that have gaps between themselves. So they should be connecting in some way. So the cell membrane has molecules that facilitate that. The physical barrier, a function of cell membrane, we already saw it is still the its nature as a selectively permeable membrane. So it's in such a way it is functioning as a barrier, but still uh, even if it is not functioning as a selectively permeable layer, it can be just a barrier. For example, certain cell membranes of particular tissue types are are designated to function in terms of protecting particular intracellular components. So certain tissues, for example, keratinized cells, for example, the skin has some tough type of tissue, as we shall see. So it is the plasma membrane that is modified to act as a barrier in such a way. So whichever antigen, foreign material that you try to invade the body not easily be allowed because the cell types in those tissues does not allow these foreign materials to easily access the internal components. So those are the functions of the plasma membrane. So we also have a function in terms of transportation. The cell membrane, we have said it acts as selectively permeable membrane. So in carrying out such a function, it allows transportation of particular uh, materials or molecules in and out of the cell. But there are different mechanisms through which those materials are transported. First mechanism is the passive way of transportation. The other one is the active way of transportation, and then the last one is the bulk mode of transportation. Remember, as we are trying to allow something into the cell or take something out of the cell, that something can be in form of a solute, that something can be in form of a solvent, or that something can be in form of a solution. So we have those three different forms of molecules. Molecules as solutions, molecules as solvents, molecules as solutes. So depending on the nature of that molecule, in relation to those three classes that we have listed, solution, solvent, and the solute, all are independent and they will undertake a different mechanism of being transported across the cell membrane. So to start off with passive transportation, this is the movement of substances along, it is along a concentration gradient. So we have a gradient where on one side we have a high concentration of this particular substance, and then on the other side we have its low concentration, but we need it to cross a particular uh, barrier, which is now the cell membrane or the plasma membrane. So that barrier, on one side, it is allowing the high concentration, and on the other side, we have a low concentration. But on the side of a low concentration, we need these substances, which might be in form of molecules, to cross this barrier to go to the lower, to the side where we have a lower concentration. So the modes of passive transportation, we have four major modes, that is simple diffusion, facilitated diffusion, bulk filtration, and then osmosis. They will apply to particular types of categories of molecules. Is it a solute? Is it a solution? Is it a solvent? If it is a solute, usually it will be the simple diffusion technique or the facilitated diffusion technique or mechanism of transportation that will be utilized by the cell membrane to transport that particular type of solids. Uh, Bulk filtration will apply to molecules that are in form of solutions. So we need uh, to transport molecules in form of solution. They are maybe they are concentrated more in the intracellular component, so we want to transport some of them balance out their concentration between the internal and the external compartments of the cell surrounding the environment. So they transport it to the side where we have a lower concentration, but it is in form of solution. So the cell membrane will utilize the mechanism of bulk filtration. It shall filter those molecules. For example, this technique can be seen in the kidney, the glomerulus, where we have filtration taking place if it is a solution. Where we have solvents, we want to dissolve a particular solid on the other end where we have a low concentration of this solvent type of molecules. We we shall see a mechanism where the cell membrane will utilize the osmosis kinds of transportation. So that mechanism, it is a solvent molecule that is being transported along a concentration gradient from one end where it is in high concentration to the other end where it is in low concentration. And you have to note that in passive transport mechanism, all these four mechanisms that we have listed down here, ATP is not required. ATP is the energy molecule, adenosine triphosphate. So we don't require these cells to produce any form of energy in trying to transport molecules passively along the plasma membrane. So the energy that the cells produce will not be utilized at any moment because this mechanism of transport, these mechanisms of transportation across the membrane are referred to as passive. You don't require any amount of energy. Go further into the the passive mechanisms of transportation. We have facilitated diffusion. This one requires no ATP utilized by the cell, but it will need a specific transport proteins. So we shall find those transport proteins in terms of protein channels, in terms of peripheral proteins, in terms of integral proteins. They are mostly in terms of channel proteins. So they are specified to transport specific molecules across the plasma membrane without the need of utilizing ATP. So we can also refer to it, we can also have an example of facilitated diffusion as carrier mediated types of transportation molecules along the plasma membrane. The other mechanism of transportation is the bulk filtration mechanism, and this one involves diffusion of both solvents and solids. So we said this one is applied to solutions. That is what we are talking about. And we are trying to highlight out diffusion of both solvents and solids. So we have a solution form of of material or substance that needs to be transported along the plasma membrane. So we shall utilize filtration. We shall first filter out maybe the solid, and then later on we filter out the solvent. It will re-interact with its solute on the other end where we had a low concentration of this particular solution, and then we still have the same nature of solution as it was before. It usually requires pressure gradient. So in filtration, we usually apply pressure on one side where we have a high concentration, forcing these molecules pass through this barrier, which is now the plasma membrane, will be received on the other end where we have a low concentration. But we are applying pressure on one side. You can also apply pressure on the side where I have a low concentration, and that pressure is acting in a way of pulling these molecules from one side where they are in high concentration, and then receiving them on this side where I have a low concentration. If you take an example of a vacuum, so a vacuum is trying to pull something from one end to the other. So those mechanisms can also be utilized, but it is usually applying pressure to the side where we have the highest concentration. So we force these molecules bypass this plasma membrane and then they go where they are in low concentration. So that is bulk filtration. So to sum up or to conclude, the passive mechanisms of transportation along the plasma membrane conclude that with osmosis mechanisms. These ones involve usually solvents. So we want a particular solvent to be transported where it is in low concentration, for example, water, and where it is in low concentration, it will perform its duties of dissolving some solid there. For example, have a high concentration of sodium chloride molecules on the extracellular side of the cell. So we need to pump water from these cells that are acting as water reservoirs. So we need to allow the osmosis to not even pump, but allow the transfer of this solvent, which is water, to the other side where I have a high concentration of sodium chloride solute molecules, so that the water will dissolve the other solids on the side where we have a low concentration of the water, which is our solvent. But it has to bypass or pass along a gradient or a barrier, which we are in this case referring to as the plasma membrane. So usually osmosis will result in a volume change on the other end. So we can balance out the volumes where we had the higher concentration, it balances the where we had a low concentration, or we can end up having a higher concentration where we had a low concentration of this solvent, depending on the amount of solute that we find on the other end. That is osmosis. So to go ahead with our mode or mechanism of transportation, we have active transport. The other mechanisms were the passive mechanisms where we had osmosis, bulk filtration, we had facilitated diffusion and simple diffusion. Now we are looking at transport mechanisms uh, that are against the passive mechanisms were along the concentration gradient across the plasma membrane. So these ones are the active mechanisms. So the major characteristics amongst all of them that we are going to see is that we are trying to transport something against against the concentration gradient of this plasma membrane. So we are trying to transfer something from where it is in low concentration and then we put it where it is in high concentration. Sometimes we need that in the body. It's not always from high to low. Sometimes we need to transport something from where it is in low concentration, then we pump more concentrations somewhere where it is already present, but we pump more. So we are acting against that concentration gradient. And if we are acting against that concentration gradient, then we need to utilize energy, which is in form of ATP, adenosine triphosphate molecules. This cell should first produce for us the energy that we need, and then we utilize that energy in form of ATP to pump or to transport the substances against the concentration gradient that has been produced by the plasma membrane, because the plasma membrane is our barrier. So we need ATP, that's a major characteristic and requirement for active transportation mechanisms across plasma membrane. It also usually requires carrier mediated ways of transportation, uh, utilizing particular transport proteins across this membrane. And you have to note that ATP will continuously be produced by those organelles known as mitochondria if it is an active mechanism of transportation. So the first mechanism of active transportation is the mechanism where we utilize ion pumps. So these ion pumps, we saw them on our plasma membrane in our introduction. We have a pump that is trying to force a transportation of certain ions from the intracellular compartment to the extracellular compartment. But we already have a high concentration on the extracellular compartment. So we are forcing a transportation to the side where we already have the concentration of that particular type of ion. So we shall utilize ion pumps or ion channels on the cell membrane for that purpose. The other mechanism of active transport is bulk transportation. Don't confuse it with bulk filtration. Bulk filtration was dealing with the solvents and didn't require any form of ATP to be utilized. But here in bulk transportation, we require some amount of ATP to be produced by this cell. And then after that ATP is available, the cell can easily transport these substances in bulk, okay, in large amounts. So bulk transportation across the plasma membrane can be seen in terms of the cell trying to release out something from the internal compartment to the external compartment. That is, for example, in releasing waste material. And that process that is utilized is usually exocytosis, something exiting uh, a particular housing. So in exocytosis, we can find that mechanism of transportation known as bulk transportation. So it is commonly used by cells that carry out purposes or functions of secretion, for example, glands. Glands secrete particular hormones. So in doing so, they utilize particular amounts of energy. And then in most cases, they utilize the energy. And then because we shall also see cases where later on we shall also see cases where there's only diffusion of these hormones. They just diffuse across a particular uh, types of cells. But in most cases, there is an energy requirement that is considered. So, exocytosis in bulk transportation mode of mechanism across plasma membrane can be found commonly in those organs that secrete, for example, those organs that secrete hormones. And this exocytosis usually it requires movement of large molecules, uh, and it is usually out of the cell because we are exiting something. So that is a depiction of exocytosis. First, there will be a vesicle that will be formed within the cytoplasm, and we call that the cytoplasmic vesicle. That vesicle contains particular little molecules within it that have been housed. So this vesicle has to fuse with a plasma membrane in some way, and then exit these components in this vesicle. So it fuses with this plasma membrane, and then we release the components that were housed within the vesicle. So that is number one. Vesicle comes near the plasma membrane, and then it fuses with the plasma membrane. So you see an interaction between the beads that were green and the beads that are purple on the plasma membrane. So they fuse together because they have a similar nature, okay, in some way. And then after fusing, excitosis takes place as the membrane now opens up externally on the external side to release the components that were inside the vesicle. And before you can see these secretory proteins being released. So these proteins can be enzymes that are required on the extracellular side, or for example in digestion. Okay? Certain glands will produce hormones that will trigger particular organs, digestive organs of digestion, to release particular enzymes to digest particular molecules that we have obtained from food. So this mode of transportation is commonly utilized in such ways. So we release our proteins that can be enzymes, can be hormones, to be received by some external receiving side. Even drugs are uh, because the cells, once they finish processing the drugs intracellularly, they have to release particular byproducts out of those processes that have taken place in the metabolizing the drug molecule. So this mode of transportation, which we call exocytosis, will be utilized that way to release the byproducts that have been produced by as a result of drug metabolism. That is exocytosis. Uh, in bulk transport, then endocytosis is the opposite of what we have seen. So there where we have seen that something is released out of the cell. Here we are seeing something being allowed inside of the cell. And there are three major mechanisms where we can allow something to enter the cell. We can do it through phagocytosis, so we have particular large molecules that are on the external side of the cell. So we undertake the mechanism of phagocytosis through engulfing that particular large molecule, then internalizing it into a vacuole form that is received, that is later received within the internal components of the cell. If we have molecules that are small in size, then we shall undertake a mechanism known as pinocytosis. Those molecules will be, will find a way of fusing with the plasma membrane, and then they are allowed into the internal component of the cell. The other way of endocytosis is uh, receptor mediated mechanisms of transportation. So a molecule on the external side fuses with or binds with the receptor that we saw earlier on, which is usually in protein nature. So receptors are usually protein molecules on the surface of the plasma membrane. So they are ready to receive a particular molecule from the external side, and then that molecule binds with them in a specific ways of bonding, chemical bonding, and then it is invaginated within the the internal components of the cell. So those are the three major forms of endocytosis. And remember, it is endocytosis is a form of transportation under bulk transportation, which is a form of transportation under active transportation mechanism. So we have a depiction of endocytosis here. So in A, we are trying to depict phagocytosis on the extracellular side, this blue side. Uh, we are forming a vacuum that is containing something that we obtained from this extracellular side. So this something is depicted as green here, and it is a large molecule. So finds a way of fusing with the plasma membrane through specific ways of chemical bonding between the two parties. Then we invaginate it into the, the cytoplasm. Part B, we are seeing small molecules on the external side. So this mode of transportation across the plasma membrane will be pinocytosis because we are considering small molecules on the external side. So that is the difference between the two. Phagocytosis requires large molecules, and then pinocytosis requires small molecules that are being allowed inside the cell. So here we are forming a vesicle, here we are forming a vacuum. That is another difference. Phagocytosis, we are forming a vacuum. In pinocytosis, we are forming a vesicle. So we can always find these vacuole and vesicle as inclusions within the cytoplasm. So this one depicts receptor-mediated ways of transportation across the plasma membrane. So we have these pale yellow structures here, Y forms of structures. They are receiving the light blue molecules here. These light blue molecules bind with this pale yellow. So this is a receptor specific for this molecule. Never bind to this red molecule. It's only specific to this blue molecule. Binds it, and then later an invagination is formed as a cytoplasmic vesicle still, and then it is processed intracellularly appropriately. So that is the end of part 2 of this series of classes about the cell, and this part was focused on the cell membrane. Be blessed wherever you are, uh, and take care.